Alright class, welcome to Lab 5, Week 3, lecture on ligation, transformation, and the selection for pNEW. And remember, pNEW is our desired plasmid with all three genes, that is ampicillin resistance, kanamycin resistance, and of course GFP along with its promoter. Alright, so let's get into it. First off, what is ligation? Ligation is that covalent linking of two DNA fragments. The enzyme, of course, is this DNA ligase enzyme, and it's an ATP-dependent enzyme that forms phosphodiester bonds. All right, so if we think about our DNA fragments, if you can think of our sticky ends after being cut with restriction enzymes. So here we have two linear pieces of DNA. One's red and one's blue, and they have both of these complementary sticky ends, right? When we mix these in a tube together, these sticky ends are attracted to one another, so these two fragments of DNA, the red and the blue fragments, anneal, right? They start to form base pairs because of those complementary pieces. Well, what ligation does, or the DNA ligase does, is it forms this phosphodiester bond between the backbone so that it covalently links the two strands of DNA. So your product is a newly recombined double-stranded piece of DNA that is now ligated together via phosphodiester bonding. All right, so that's ligation. All right, the next topic I want to go over is transformation. Now, we've talked briefly about this before, but today we're going to go a little bit more in depth. All right, so just to recap, transformation is just inserting DNA into an organism. And the DNA we're working with today will be plasmid DNA that we've recombined from the fragments of the digestion, and the organism we're working with is E. coli. So to get E. coli cells to readily accept DNA, we make competent E. coli cells. That is, E. coli cells that can readily accept DNA, right? And the way we do this is by washing them with calcium chloride and keeping them cold. All right, I'll talk about these two factors in just a second. All right, so if you think about the structure of DNA and the structure of the cell membranes, you realize that they're both negatively charged, right? DNA has that phosphate backbone, and the phospholipid bilayer has this hydrophobic or hydrophilic end that is also negative. And because of that, you're going to get this ionic repulsion. These two will not be in close proximity with each other because of that ionic repulsion, negative and negative, repelling each other, right? So how do we overcome this? If we want to get the DNA past this cell membrane, how do we overcome this ionic repulsion? Well, we treat it with calcium chloride. What happens in solution is that the calcium chloride ions dissociate, and the calcium ions, a positive ionic charge, interact with both the DNA and the phospholipid bilayer negative charges, so it sort of neutralizes that repulsion so that they're allowed to be in close proximity with each other without repelling one another. Okay, so now the DNA is close to the cell membrane. That's not enough, right? We want the DNA inside the cell membrane. So how do we get it past the cell membrane? Well, we do that by heat shocking it. Right? And it's a shock, right? It's not a gradual increase. You need to shock it to a certain degree. And what we do is we bring it from ice cold temperature to 42 degrees Celsius. And that jump in temperature opens pores within the cell membrane and allows DNA to traverse the cell membrane. And again, it needs to be kept cold because it's an extreme jump in temperature. The jump in temperature from room temperature to 42 degrees Celsius is not enough to shock those cell membranes open. You need it ice cold to 42, opens pores, DNA moves through. All right, so last week we confirmed successful double digest using agarose gel electrophoresis. That means that this week we know we have two fragments from pBag and two fragments from pCan as a result of double digestion. And keep in mind that each fragment is going to have sticky ends specific to those restriction enzymes, and that's what these blue and yellow little dots are on the fragments are supposed to represent. So this week we're going to take all those fragments and we're going to ligate them together. And remember, because each fragment was cut by the same enzymes, they all have those same sticky ends, there's going to be a number of different ligation products that we can produce through ligation. And if you remember on page 52, it shows all those different patterns like K1, K2, B1, B2, etc., etc. And all those are plausibly produced from ligation. Okay, so we're going to take all those ligation products and we're going to transform them into those competent E. coli cells. And that means each E. coli cell could take in a different one of these plasmids, right? But the only plasmid we want to keep investigating is the plasmid with our desired genes. That is ampicillin resistance, kanamycin resistance, and GFP. So the way we select for our plasmid is to grow these E. coli cells on an agar, an LB agar, a nutrient-rich agar with ampicillin and kanamycin. So if E. coli can grow in the presence of ampicillin and kanamycin, that means it has our plasmid of interest. So if it grows, it has our plasmid. So if it grows on ampicillin and kanamycin plate, that means it has both ampicillin resistance and kanamycin resistance. We also need to check for GFP enzyme, and we do this by shining it with UV light. If it fluoresces green, it has the GFP enzyme. All right, one more thing I want to talk about are the controls for this experiment. We're going to use these controls to help us troubleshoot if something goes wrong in the experiment. That is, if we don't see any green colonies on the ampicillin and kanamycin plates, we can look back at our controls to assess what may have gone wrong. And we're going to use two controls. We're going to use no DNA and control DNA. And the no DNA means no foreign DNA transformed, used to transform E. coli. So it's just going to be our competent E. coli cells spread onto LB agar, the nutrient-rich agar. And the growth or no growth of these E. coli cells will allow us to assess the viability of these E. coli cells. That is, can these cells even grow, right? And the control DNA contains or is this plasmid UC18, PUC18, and it contains an ampicillin resistance gene. And we're going to take this control DNA and we're going to transform it into E. coli and then spread that E. coli into an LB agar with ampicillin. And now the growth or no growth of our control DNA will allow us to assess the transformation protocol. So if we don't get growth on this plate with the control DNA, then maybe something's wrong with our transformation. If we don't get any growth on our no DNA, maybe something's wrong with these E. coli cells. So these are what we'll use to troubleshoot if something goes wrong. All right, that's it for this week's video. Hope you guys learned something, got some insight on what's going on. If you have any questions, please submit in the comments below. If I made a mistake, please be more than welcome to call me out, and I will see you guys next week. Thanks.